And thanks very much for inviting me to share this with everybody in the PANGEA group. So first of all, it's really important, of course, to acknowledge that I am speaking on behalf of an enormous team and an enormous piece of work funded by our partners at NIH, PEPFAR and Bill and Melinda Gates Foundation through 3IE. And the slides I'm going to talk to today are based on the presentation that Richard Hayes gave at the CROI meeting earlier this year. And we are still hoping, this will be published in the next month or so. So I hardly need to describe this to the audience here, but the background clearly is based on the data we all know very well about how an undetectable HIV viral load reduces significantly the risk of HIV transmission. And we get to this U equals U, undetectable equals untransmissible, from the data from the HPTN 052 and the more recent partner study. So that on an individual couple level means that if the individual who's living with HIV is on antiretroviral therapy and their viral load is undetectable, they will not transmit. But obviously at a population level, it's key that everybody knows their HIV status to allow all those people living with HIV to actually get onto treatment and suppress virus, which for most current regimens takes several months to happen from starting treatment. And the modelers, initially from work from other modelers and then WHO, suggested that if we could test everybody within a population and treat everybody living with HIV successfully so their virus is suppressed, we could achieve very significant reductions in HIV incidence. And again, familiar to most of the people in this meeting, previous trials that have proposed different models of delivering a UTT approach at a community level have shown somewhat different results. So the SEARCH trial that was led by Dan Havler and group, undertaken in Uganda and Kenya, as well as the TASP trial run through the ANRS and the Africa Centre in KwaZulu-Natal in South Africa, in fact showed no significant impact comparing their universal test and treat approach versus standard of care. And it may well be that although they got very good coverage in both studies of knowledge of HIV status, in the SEARCH trial their control communities or standard of care communities also received quite significant interventions, which may well explain why they found no marked difference between their two study arms. And the TASP trial, whilst they got good coverage of knowledge of HIV status, did not significantly impact the ART coverage in both study arms. So again, these are explained by findings that have subsequently reported from both studies. The Botswana study, which again reported at the Amsterdam conference last year, found a borderline significant reduction in incidence of about 30%. So three very enormous, very important trials, but have not shown conclusive agreement with the initial modelled impact of delivering universal test and treat. So PopART was run in parallel with many of these studies. It's the last one of the four main community randomized trials to give an answer. And our question, like these other studies, was basically, can universal test and treat be delivered in practice in generalized epidemics in sub-Saharan Africa? And if we can do this, what impact on HIV incidence can be achieved through these pathways? So this slide summarizes the study design. And in PopART, unlike the other trials that I've just mentioned, we had three study arms, and this really reflects what was happening at the time when the study was first begun. And at the time when the proposal first went in, antiretroviral therapy was initiated based on a CD4 threshold of less than 350 cells, and therefore we decided that we would look at a community-wide intervention where antiretroviral therapy was commenced irrespective of CD4 count, which is Arm A, which at the time this was begun was well outside of guidelines. There was not the data we currently now have from START and Temprano. And so this was perceived to be an experimental approach to treating people living with HIV. Arm B, at the time it was designed again, was different in Arm A only in the ART initiation threshold. So in Arm B, they had exactly the same community intervention, which I will describe shortly, but ART initiation was only given according to national concurrent guidelines, which, as I say, at the time was a CD4 threshold, which has subsequently changed throughout the period of the trial. And then Arm C communities had what was current and changed over time standard of care. So all ART provision and HIV testing was delivered through standard government facilities, and testing was mostly run through those facilities, although in some communities there were some campaigns. So PopART was an enormous trial, and the entire study population we estimate to be around about 1 million people. The primary outcome measure of the study, of course, is HIV incidence. And in PopART, what we did, which is different from some of the other study designs, was we randomly recruited a population cohort of individuals who are living within those communities, who obviously consented to be part of the research cohort and were aged between 18 and 44. These individuals were followed up annually over 36 months. So in total, they actually had 4 study visits around about 12 months apart. And we aimed to recruit 2,500 individuals from each community. So PopART included 21 communities, 7 communities in each of the study arms, A, B, and C, as you can see here, 12 communities in Zambia and 9 in South Africa. And you can see that they are quite spread across Zambia and in South Africa, all around the Western Cape, around the city of Cape Town and in the winelands. So the exact details of the intervention, which just to remind you, was only offered in all Arm A and Arm B communities, was a novel cadre of staff who themselves were lay community residents, and they were specially trained to deliver HIV counselling and testing services within the households. They also offered VMMC referrals and facilitated HIV negative men to get to standard government VMMC services. They encouraged and referred pregnant women who were living with HIV for PMTCT services. They offered STI screening for symptoms and TB screening with samples of sputum collected and delivered to the clinics for those with a cough productive of sputum, and they offered male and female condoms. And one of the factors that I think has been very successful within the communities but took some time to develop was establishing the acceptance of these individuals. So most of the people who trained as CHIPS workers, and in total we had about 600 CHIPS altogether in both countries, they worked in pairs. Where possible, we tried to pair male and female workers, but we had more CHIPS workers who were women than men, as is often the case in healthcare workers in these communities. And they wore a recognised uniform so that within the communities they were identifiable. And they would repeat door-to-door delivery of various different aspects of the household intervention. They returned to different households if they'd missed participants or household members were not resident. They trialled different working shift patterns to work in the evenings and at weekends. And based on an initial census, we had some sense of how many people were resident in those households. HIV testing and all of the services were offered to every single individual who was present within the household, irrespective of age. And even if people were just staying within the household for a short period of time, they were also offered HIV testing. The CHIPS teams collected information on an electronic data capture device, just in terms of HIV testing and uptake of treatment. We reviewed some of these on a monthly basis to try and adapt as we were working through the intervention, people we were missing, groups that we might have been missing, and to see if we could optimise how we could do this a bit better. So one of the key differences before I get to the results is the difference between Arms A and Arms B. And this timeline shows you at the top line how we delivered the CHIPS intervention. The CHIPS are what we call our community HIV care providers. I've just explained what they did, and they worked in what we called annual rounds. They weren't exactly on a yearly basis, but approximately so. And they delivered repeated HIV testing and services on a yearly basis to every household within their allocated zones. The population cohort, just to be distinct, is those individuals that informed our primary outcome. And this is the research cohort who were visited, where blood samples were taken and a questionnaire was asked at annual study visits. The ART eligibility for Arm A, as you can see in the pink bar, was universal access to treatment throughout the entire study period. But importantly, for Arm A communities where the attendants at the clinic identified people who had a CD4 count above what was the prevailing guidelines, that person had to sign a written informed consent to say that they understood that it was considered experimental, that they were starting treatment outside of guidelines. And this is an important consideration, possibly to explain some of the findings, which we'll talk to you about shortly. In arms B and C, as the guidelines have shifted to universal treatment, and this is the second to bottom line across, you can see in Zambia, universal ART irrespective of CD4 count was offered according to guidelines, and therefore no longer required consent to be taken from the beginning of 2016. And in South Africa, it was slightly later, towards the end of 2016. The important third line, where it says the primary analysis period, is the period of time within which we looked at HIV incidence. And as you can see, for the majority of that period of time, when we look at the results, you can note that individuals accessing the clinic were all being offered universal treatment during most of that period, between arms A, B, and in fact C as well. So how did we select our research cohort? Individuals had to be resident within a randomly allocated household. They had to be between the ages of 18 and 44 years. And within that household, a research assistant would gain entry. They would identify all people who lived there. And then there was a second random allocation of a consenting resident who was within that household and who was within the age range. People from the research cohort who consented had one blood draw every year. This was a sample that was stored for HIV, HSV, HIV viral load, and viral sequencing. And they were asked various degrees of complexity of sexual behavior questionnaires, whether they had access to voluntary male circumcision, their knowledge of HIV status, and ART for self-reporting HIV positives. There was a raft of economics questions and a quality of life questionnaire. So during the study period, we have called each annual visit PC0, as in the baseline. PC12 is the first 12-month visit. PC24 is the subsequent, and PC36 is the final research cohort study visits. So initially we enrolled 38,477 participants. We then went back to try and find those original individuals in the subsequent visits. And what is here is just showing the final follow-up. Terminated means that for various different reasons, individuals were not continued in the cohort. And because these PC participants were meant to be our view, if you like, of what was going on in those communities, if they had moved away outside the community, even if they happened to be available or consented to take part, we did not continue to follow up. So we only retained individuals who still were resident within our communities and who obviously continued to consent for the study. In certain communities where we felt that we had lost a large proportion of or a higher proportion than we had wanted to of our individuals, we enrolled two further groups, which we've called PC12N and PC24N, which were also contributing to the period of the primary study endpoint. So this table just identifies the baseline characteristics of the population cohort, and they're divided by arm. You can see on the top it's arm A, arm B, and arm C, and we recruited around about 13,000 individuals into each of the study arms. The age, as you can see, was spread reasonably as expected between the age ranges we talked about, between 18 and 44. And one of the things that's quite significant is we enrolled far more women than men. As expected in these communities, the prevalence at baseline overall for HIV was almost double in the women compared to the men. But importantly, for each of the study arms, there was good matching. And just to note for future reference when we talk about it a bit later on, the baseline viral suppression, which is viral load measurements taken on HIV positive samples stored in our central repository lab at Hopkins, showed about 56% of individuals were suppressed on antiretroviral therapy, which actually is quite comparable with the latest Zamfir data that's been published. Medical male circumcision at baseline was actually very limited amongst most of the HIV negative men. This is self-reported male circumcision, but again, comparable across study arms. So how well did we do at actually delivering the intervention? This slide summarizes an enormous amount of data, but it's based on the antiretroviral coverages in Arm A and B communities at the end of the trial. So this is the data that we have captured from the CHIPS teams. This is not from the research cohort. This is how many of the people living with HIV, when they attended the household visits, tested for HIV and were on antiretroviral therapy. And then it's broken down by men and women, and also broken down by Arm A, which is in the squares for men, and blue squares. Arm A is in black and Arm B is in blue. And if you look at this along the bottom, you can see that actually from the age of approximately about 35 onwards, we do pretty well in both Arm A and Arm B, and amongst men and women for HIV positive individuals within the communities reporting that they are on antiretroviral therapy. And consistent with many other studies, we can see that where we really fall below the line is in the younger people, certainly between the ages of 18 and 35, and lower levels in men in the squares and the women in the circles. But again, you can see that actually there's not a significant difference between the Arm A communities and the Arm B communities in terms of ART coverage. Just to be very clear, and it may seem a very obvious point to make, but I know sometimes, particularly people who are seeing this for the first time, it can be slightly sort of confusing. We do not have parallel data for Arm C communities here because, of course, they did not receive the CHIPS intervention. And so the data I'm showing you here is from the CHIPS, not from the research cohorts. So the study primary outcome, we measured HIV incidence from blood test result from the population cohort. We pre-specified that the time period we were going to look for was between PC12 and PC36. So that's HIV incidence between months 12 and 36 after study enrollment. And that was based on the assumption that for most people who are newly diagnosed with HIV in the PC0 phase of the study, it would take some time for antiretroviral therapy to successfully suppress viremia. We took the time of infection imputed for seroconverters who were not seen at PC12 and or PC24. And we looked at the impact comparing arms A versus C and arms B versus C. And we used the method standardly reported for matched cluster randomized trials. So this slide is a summary of the main trial results. And if you, just to orientate you, so each triplet has been matched in the first place, and they are numbered in the different colors you can see at the bottom. So for example, in the red line is Zambian triplet 1. And for Zambian triplet 1, there is an A community, a B community, and a C community. So on the left-hand panel, you will see a Zambian community 1 in arm A, the red line, compares the incidence over time in the study period we're looking at compared to incidence in that same triplet in arm C. And overall, you can see the first comparison is all the triplets of arm A versus arm C to show that whilst 4 out of the 7 triplets, there is a reduced incidence of HIV in Arm A compared to Arm C matched triplets. In actual fact, in 3 communities, the lines, if you like, go the wrong way. The incidence in Arm A is either the same as Arm C or higher. On the panel on the left-hand side, in exactly the same way, by matched triplets, Arm B communities are shown compared to Arm C communities in terms of incidence. And you can see here there is a perfect line where all of the Arm B communities show a lower incidence compared to the Arm C communities. How we achieved this result, just to go through the numbers, the primary outcome was taken from 39,702 person years. There were 553 incident HIV infections, and I'm going to show you in detail now about how this is actually broken down in terms of numbers in the subsequent graphs. I've included this figure here just to show you what happened just looking at the Arm C communities, because of course if you look at the background changes in incidence, there has been some reduction in incidence in Arm C over the study period. And remember, all of our Arm C communities received standard of care at the clinic. So whatever we observed in the figures you have just seen, Arm C communities also had a reduction in incidence over the time of the trial. And so our reduction in incidence, in fact, is on top of the reduction in incidence seen in Arm C communities. So for the primary analysis, you can see here, presented by Arm, the adjusted rate ratio for Arm A versus C is 0.93, so a 7% reduction in incidence and not statistically significant. However, Arm B communities have shown adjusted rate ratio of 0.7, 30% overall reduction in HIV incidence compared to Arm C, and a p-value of 0.006, so highly statistically significant. And we were very surprised when we first saw this result. In actual fact, we wondered if by mistake we'd got our arms muddled up, but in actual fact this study analysis, as you can imagine, has been repeated on multiple times by various different very senior statistical groups, and the same result has been found. So now we need to understand what we have found and what this means. And I was just thinking through, just to be clear, what are the key differences between Arm A and B communities? A, B, and C. So in Arms A and B, very little difference in that they both had CHIPS teams. In Arms A, right from the very beginning of the trial, as I've mentioned earlier, they were offered antiretroviral therapy right from the start, irrespective of CD4 count. But until the guidelines were changed, individuals living in Arm A communities had to sign written informed consent. Now, when we went back to have a look at this, there were very few individuals who actually declined to sign written informed consent and accept antiretroviral treatment by the time they'd reached the clinic. In the Arm B communities, of course, they received the CHIPS intervention in terms of the household offer of testing and linkage to care, but they were only offered antiretroviral therapy at the local clinics according to the prevailing guidelines. But they also did not have, therefore, any requirement to sign any consent before starting antiretroviral treatment. So essentially, in fact, very little difference overall in terms of the impact on the measures I've just talked to you about in incidence between individuals in Arms A and B, but clearly we did see a difference overall in the impact on incidence, and that's been quite difficult for us to explain. One of the obvious ways that might explain it is to look at viral load suppression. And we have data at the moment just from the PC24 study visit, which has shown in this table here. So this is taken from blood samples collected from the PC participants at the 24, so that middle study visit. Sorry, there's a lot of noise going on and I don't know quite what that is. To show you viral load suppression, looking at the different study arms. So bearing in mind, if you remember what the baseline viral load suppression was by study arm before we started, which was around about 54%, you can see here that by 24 months after the intervention, in arms A and B, there's been a dramatic improvement in viral suppression, and particularly compared to arm C, which you can see here in the red. So 16% increase in viral suppression in arms A versus arm C, and 8% improvement in arms B versus arm C. Now, interestingly, we have also seen an improvement in viral load suppression in arm C compared to baseline, which again could reflect changes in guidelines and may also reflect the fact that our PC participants by this stage in the trial have also been talked to about HIV, been encouraged to test for HIV, and in fact the PC participants, although they did not receive the results of the venous blood samples they were taking for research because the testing was done retrospectively, they were offered a point-of-care finger prick test for HIV. So we may have seen a Hawthorne effect here, that we actually improved outcomes in our observed viral suppression in arm C because of them being part of the study trial. If we look at this by study year, again, this is showing the same data on viral suppression in HIV positive PC participants by each study year. And you can see by study year that we achieved really quite sustained viral suppression, in fact, even a slight increase between PC24 and PC36. So by the end of the trial, in arms A and B, we have certainly reached the UNAIDS 1990 targets of 73% viral suppression, and in fact almost reached that in arm C as well. We then had a look at whether viral suppression correlated with HIV incidence, which of course one would expect it would. And using data from PC24, which is in the middle of the primary analysis period, overall viral suppression in arms A and B was about 72.7% and arm C was 63.8%, the table you have just seen. The corresponding proportions, therefore, that were virally unsuppressed were 27.3% and 36.2%, which would imply a reduction in HIV transmission of around about 25%. We did do a post hoc analysis. We compared arms B and C versus combined arms A and B and compared this with arm C communities, and we showed again a significant reduction in HIV incidence of around about 20%. So you can see that, as one would expect, there is quite a nice relationship between virally unsuppressed at a population level and reductions in HIV incidence. So to summarise our key observations, I think we feel this trial has been an enormous learning, and we've not only achieved our primary goals, but there is a vast amount of wealth of work that I haven't even begun to talk to you about in terms of the community mobilisation, community engagement, stigma, a lot of social science embedded in this, and Christoph's group are working with us on the phylogenetic analysis, which may well explain some of the observations I've just presented to you. I think we feel we have shown that it is feasible and acceptable to deliver community-wide HIV services in these large urban communities in Southern Africa through this system of community HIV care providers, so that every community resident is visited and provided with similar services every year. The trial results on HIV incidence, together with the data on viral suppression, show quite convincingly, we feel, that the CHIPS intervention can be implemented and could be expected to reduce HIV incidence each year further by around about 20%. And we hope this will build up year on year as the intervention is continued to be offered. You saw from our graph that although we've done a good job, we still could do better, and the coverage of the CHIPS intervention in terms of knowledge of HIV status and coverage of ART is lower amongst some subgroups, and this is in agreement with many other studies as well, in particular amongst men and young people. And continued efforts really do need to be looking at how we can fill these gaps and to get a better coverage to every subgroup of the population. So in summary, we have achieved the first two UNAIDS 1990 targets in our arms A and B. We saw high rates of viral suppression, which significantly improved over the study period. PopART with treatment according to the local guidelines, so our arm B communities showed a significant reduction in HIV incidence by about 30% in these very high burden settings. But it's quite daunting to still see that they're extremely high rates of incidence despite all of this intervention. The lack of the overall effect in the full intervention arm A communities is unexpected. And at the moment, we don't really have a neat explanation to look to why this may be happening. We were surprised, and I've shown you data to show that actually it was not because the people in Arm A communities do not accept antiretroviral therapy, and in fact the viral suppression data shows that the Arm A communities overall had a slightly higher rate of viral suppression compared to the other arms. We're doing a lot more analysis of the quantitative, qualitative and phylogenetic data to explore and explain the findings. But I think our key message for policymakers is that a community-based service to achieve a universal HIV testing and linkage are the key components of combination prevention in the global effort to achieve HIV control. And I've mentioned it at the beginning, but this has been an enormous piece of work, and I have been very privileged to work with a fantastic group of people in both countries, both all the implementation of the treatment and the MMC services were supported by PEPFAR and delivered through government facilities. And the other study PIs which have led all this work over the last almost 10 years, in particular, I'd just like to acknowledge Richard Hayes, of course, who's the study overall chief investigator, Helen Ailes, who led all the work in Zambia, and Nalda Bears and Peter Bock, who lead the Desmond Tutu TB Centre in South Africa. And then, of course, all the implementing partners and government agencies that delivered the intervention.